So we're going to begin our webinar today. Welcome and thank you for attending our Ocean Acidification and Harmful Algal Bloom webinar series. My name is Maggie Broadwater from the National Centers for Coastal Ocean Science, and I'll be moderating the webinar today. Jennifer Mintz from the NOAA Ocean Acidification Program will be our producer, working to keep Adobe Connect going. If you have problems with Adobe Connect, please enter it into the chat box on the upper right corner of your screen, and we'll try to address them. We were having some issues with the closed captioning today, but a transcript will be available with the recording following the webinar on the webinar website, which we will provide at the end of the webinar. This series is laying the foundation for a virtual workshop on August 11th through 13th, where we will discuss harmful algal bloom and ocean acidification interactions, the major questions surrounding them, and what research priorities can help address gaps in our understanding and management of these factors. This is the second in a series of webinars to prepare for our virtual harmful algal bloom ocean acidification workshop. The workshop will help us to identify priorities and research products that could be incorporated into a future request for proposals on the intersection of harmful algal blooms and ocean acidification. If you are not yet signed up for that workshop but would like to participate, please contact Maya Sharp, whose email will be provided in the chat box. Now I'm going to let Maya introduce herself, and she'll share a little bit about her summer project with us. Maya, are you on? You may be on mute. So we may be having some technical issues. Oh, I see Maya came off mute. Are you there? So I will tell you about Maya's project today, just in case. She will be completing a project based on the content of the webinars and the questions and discussions that follow. The chat box will be monitored and recorded during the webinar, and there will also be a Google form that we will use to inform our workshop direction and future directions of research funding. Those accessing the webinar online will have the opportunity to comment and ask questions through the Google form, which will be a link in the chat box. And Maya will be compiling these and synthesizing them to provide to workshop participants in formulation of recommendations for future funding priorities. And Jennifer Mintz is going to tell us a little bit about Adobe Connect logistics.
Hi there. So just a few logistics. Audio, as you all may be aware, is over the computer, so you can listen through computer speakers or headphones. If you are having trouble hearing, you may either turn up the volume, and you can do that, of course, on your computer or headset. You can also turn up the volume in Adobe Connect. So in the top menu bar, you can pull the click down arrow to the right of the speaker icon and select Adjust Speaker Volume, and do so there. If you're on a headset, you go to that same speaker icon, hit the pull down and select the speaker if you'd like to listen to a particular headset. If you are still having trouble hearing, please log off and log back on and try entering Adobe Connect using the application rather than the browser. And we hope that will allow you to both see and hear the presentation today. To view our presentation or our presenters' slides today, you can view that in using the full screen mode, and that's the four arrows in the top of your slide there. Those are in the upper right edge of the slides. This button toggles to make the slides larger or smaller, and you will, just a note, lose the chat and captioning or lose the chat box in full screen, but of course can toggle back to a regular view at any time to ask questions or add comments in the chat box. So we do ask that you enter your questions, any questions you have for our presenter, into the chat box in the upper right-hand corner of your screen. We will hold questions until the end of this presentation. And also note that we will be sharing any relevant links that our speaker mentions into this box as well. As Maggie mentioned, we are asking that all participants support Maya in her summer project and take a few moments to fill out the questionnaire. That is a Google form that is a link in the chat box at the end of this presentation. At the end of today's presentation, we will be opening it up for a discussion, and if you'd like to comment but we're not the original questioner, we just ask that you raise your hand. Again, that icon is in the top menu bar, and you can click that icon and we will unmute you so that you can contribute or ask any further questions that are part of the discussion. We will be posting the recording and a PDF of the slides on our workshop webpage, which will be in the chat box momentarily, a link to that. And once again, a link to that questionnaire will be on the website for viewers to send those follow-up questions and comments to our presenter and the workshop organizers after the webinar. We really encourage you to fill out this questionnaire, as they will really help inform and build the foundation of our discussion at this upcoming virtual workshop in August and inform the directions of future research funding. So with that, I will hand it back.
I am very happy to share that today's speaker is Dr. Raphe Kudela, who is a professor of ocean sciences at the University of California at Santa Cruz. Dr. Kudela has conducted research on aquatic ecology for more than 2 decades with an emphasis on marine systems, but also including the land-sea interface and freshwater systems. His research focuses on the factors and processes linking phytoplankton productivity to higher trophic levels, including the ecology, mitigation, and prediction of harmful algal bloom events, changes in global productivity and fisheries, and linkages to human use of aquatic systems. Dr. Kudela has served as the chair of the Global Ecology and Oceanography of Harmful Algal Blooms, or the HAB program that's sponsored jointly by SCORE and the Intergovernmental Oceanographic Commission of UNESCO, and as vice chair of the Global HAB International Science Program that continues the GeoHab legacy. He is an ex officio member and a former co-chair of the United States National HAB Committee. Within the Ocean Observing Framework, he serves on the Executive Committee for the Central and Northern California Ocean Observing System, also known as Synergies. Today, Dr. Kudela will talk about synergies between ocean acidification, hypoxia, and harmful algal bloom networks, using California as a case study. So welcome, Raphe, and thank you for sharing your presentation with us today.
All right, thanks so much. And so it's a great pleasure to be here. There's my slides. All right, and so I'm going to talk specifically about how HABs and OAH intersect in California. And so I'm following Chris Gobler from the first presentation, and he gave a really fantastic overview of more of the global drivers. And I'm going to really focus on California. And so I may leave out references or papers or studies that were done elsewhere, and that's deliberate. I'd like to acknowledge before continuing, some of the data I'm showing are from my colleagues, Daniele Bianchi, Faisal Kasturi, and Pierre Danian at UCLA, and then some of the modeling that I'll allude to briefly, it has been led by Clarissa Anderson at SCOOS. And lastly, I'd be remiss if I didn't say that much of the work I'm presenting has been funded by EcoHab, MerHab, PCMHab, AITT, and it's embedded within both SENCUS and SCOOS within the IU's framework. And so jumping right in, you're all probably very familiar with this paper. This was published by Mark Wells a few years back, talking about the stressors and interactions that are going to drive changes in HABs in a future ocean. And Chris also touched on this in his first presentation. And of course, OA is in there on the right as pH and PCO2, but there's others as well. And so we've got changes in temperature, changes in nutrients, changes in stratification. And just a reminder that this is a complicated problem, and that we can focus on some particular aspects of this, but it's really happening in an environment that's changing multiple drivers at the same time. And so again, you're all probably super familiar with this figure, but I just want to point out this is the Keeling Curve, and Chris pointed out that as of May, we're up to 417 parts per million. I want to draw attention to the pH, and so the pH is also dropping here in the blue line. We're seeing the pH from 1988 to about 2018, and we're seeing this gradual decrease as the carbon dioxide increases. And what I want to point out is it went from about 8.1 down to about 8.0. And so those are, at the global scale, the sort of pH changes that we're seeing. Here in California, we of course also see the effects of climate change and acidification of the oceans. This map is showing the depth at which aragonite becomes undersaturated, and you can see a couple of things. One is the numbers that you're looking at are showing the depth where it occurs, and they're relatively shallow along the coast of California and the entire west coast of North America. And that's because we have intense seasonal upwelling. And so we're bringing up what's already acidic water right up into the coastal zone. And of course, that's also where things are interacting and driving harmful algal blooms. But again, as a reminder, this is all taking place in the context of other changes as well. And so here is June to August 2015, looking at temperature. And we see that this is the anomaly showing that the entire West Coast experienced a marine heat wave as well as an El Nino. And so these changes in OA are being driven in part by changes in that deep water circulation, but they're also interacting with these changes in temperature. And one of the things we're really interested in, of course, is how does that manifest at higher trophic levels? And so from that 2015 time period, everywhere you see a colored dot, we detected fairly high levels of domoic acid in fish viscera. And so that's really the interest, is how are temperature and ocean acidification and these other drivers manifesting as potential ecological problems. And so again, here in the California current, we, of course, are experiencing changes in temperature and ocean acidification. At the top, these panels are just showing 1750, 2005, basically today, and then 2050. And as the rest of the ocean is changing, so is the California current. And we are seeing gradual decreases in the long-term pH of the ocean. These panels down below, this was a really nice paper that was just published by Brady and colleagues, demonstrating both the long-term pattern and the obvious seasonality that we have because of the upwelling cycle on our coast, but also that models are fully capable of predicting these changes in pH and ocean acidification going from the 1990s to today. What I want to point out, however, is again, we're talking about changes in pH that are going from about 8.13 to maybe 8. And so on a global ocean scale, these are still pretty small changes in pH. As we zoom in and start to look at event scale, what we see is California current and eastern boundary current upwelling systems generally are really variable. And so here what we're looking at is a nice analysis that was published by Francisco Chavez and some others looking at PCO2 and pH. And I just want to highlight, if you look down here at panel E, PCO2 in a matter of a few days in a relatively small area, so we're up in the northern part of Monterey Bay, is going from less than 400, so below atmospheric concentrations, to more than 1,000. And so we already see in the California current on the event scale PCO2 levels that are not going to reach that concentration globally for many, many, many years. If we look at the pH, we also see that our pH can go as high as 8.2 or 8.3 and as low as 7.6 on these sort of event scales. And so there are places in the ocean where it's a natural experiment, where we're already seeing these wide ranges in pH and PCO2, as well as wide ranges in nutrient loading, wide ranges in temperature. And so looking at California, and again, a reminder, I'm going to focus primarily on California. But if we look at California, there's many potential HAB organisms that we're interested in. I am going to today focus primarily on Pseudonychia because that's the one that we have most information on. but we do know there's pH effects on other organisms such as cyanoHABs, where we do see a strong correlation between alkalinity and the presence of toxins from cyanoHABs. And so Chris introduced this really nice review paper by Brandenburg et al. That came out last year, 2019. And one of the main points of that paper is that harmful algal bloom species generally seem to have a competitive advantage compared to non-harmful algal bloom species. And I'm going to be a little bit of a naysayer, and I'm not saying there's anything wrong with that analysis. I think it was a beautifully done paper, but I will point out that for the organisms that were on the West Coast, that meta-analysis only included 3 sets of experiments. There was one on Alexandrium, one on Pseudonychia multiseries, and one on Pseudonychia fraudulenta. And so that's the totality of data that's relevant to the West Coast that was in that review paper. And so we really have not a lot of data to discuss these sorts of things. Why is that important? I'm going to give you an example using temperature, which we have a lot more data on. This was a paper that was led by Phil Boyd a few years ago, and what we did is we got together a group of people that had cultures in their lab, and we decided on a standardized set of experiments. And we all conducted the experiments with our species at the same time using the same methods. And you don't need to worry about the details, but what I want to show you is the red in each of these panels were the data from our study, this sort of community study, and the blue are the data that you would find in the literature. And so on the left, we're looking at niche width, the temperature that these organisms can tolerate, and on the right, we're looking at the specific growth rate. And my point here is that if you just simply went to the literature and grabbed some data, versus redoing the experiments yourself, we get widely varying responses to temperature when the experiments are all done the same way with the same methodology. And so it's a reminder that we all know that just because it's published doesn't mean that it's representative of what's going on in the ocean. And so we can show that pretty easily with temperature because there's a lot more data. I think as we accumulate more experiments on ocean acidification, we're going to start seeing the same thing, that there's quite a bit of variability in the responses that we're actually seeing in organisms in the ocean. And so how do we get around that? One way to get around that is to take advantage of all the data we're already collecting. And so here on the left, I'm showing you between Senkus and SKUs, we have a series of long-term time series where we're monitoring harmful algal bloom species and toxins and nutrients and other parameters. And on the right is the ocean acidification data that is part of a Pacific consortium. And so Senkus, SKUs, and also NANUs, which is the IU's group that's responsible for Oregon and Washington. And one of the things I want to highlight is there's a lot of synergy here. Many of these data sets are co-located, and so where we have HABs, we also have pH. Many of these sites have, or a few of these sites have percolators, but it's not necessarily well coordinated in terms of science. And so we're collecting all these data within the framework of IUs, but they're not well coordinated, and so it turns out to be challenging to actually say, well, is there a direct link between HABs and ocean acidification? Because even though the data are being collected by the same groups in the same places, they're not really being used for the same purposes. But we have a lot of data that we can start to look at. And so one way to look at that, I'm going to introduce this idea of something called a statistical method that uses cumulative probability analysis. And what this does is if you have sufficient data, it can statistically identify thresholds that can identify, for example, where you might be concerned about impairment. And so as an example, I'm going to draw from Satula et al. In 2017, looking at San Francisco Bay. And here the question was, is chlorophyll a good indicator of impairment, of poor water quality? And so on the left, we're looking at chlorophyll increasing on the x-axis from a very long time series, and on the y-axis, it's the probability that you would have a harmful algal bloom defined as exceeding a threshold in cell counts. And what we see is San Francisco Bay, sort of its background threshold is we have about a 30–35% chance of seeing a HAB regardless of the chlorophyll concentration. But as chlorophyll increases, HABs also increase, or the probability of seeing them increases. And we see that you cross the 50% threshold somewhere between about 15 and 25 micrograms of chlorophyll. We did the same analysis with dissolved oxygen. You get the same sort of pattern. We see that as chlorophyll increases, the probability of having a low DO event also increases, and the chlorophyll is remarkably similar. It's somewhere between about 10 and 15 micrograms of chlorophyll would be where you need to start being concerned about what's going on in San Francisco Bay. And so using that just sort of as a background of how this analysis works, some of you may have seen this presentation, but we've gone in and we've tried to anchor that sort of analysis in field data looking at pseudonychia and domoic acid. And so given these really large data sets, can we actually infer what's going on from the field data and then compare that to the laboratory experiments? And so here we combined a fairly large data set of domoic acid concentrations from the west coast of the United States, some basic statistics. We have about 10,000 observations. We have other parameters such as temperature and salinity and nutrients. And we applied that cumulative probability analysis. And what we got is this, where there's a peak probability. It's more likely you're going to have domoic acid at a temperature of about 12 degrees Celsius, which, if you're familiar with the west coast of the US, that's basically upwelling temperatures. But what it also showed, which was really intriguing, is if you get above temperatures of about 20 degrees Celsius in the field, we simply do not see domoic acid. And so this is somewhat contradictory to some of the literature that's been published that suggests that a warming ocean is going to result in massive increases in toxicity for groups like pseudonychia. When we look at what we actually see in the oceans, at least in our current ocean, we're not seeing that. And so I'm personally a big fan of alternating between lab experiments where you can really control and look at the physiology, and then taking that back into the field to see whether reality actually matches those lab experiments. And so that was an example using temperature. But where I'm going with this is we can also do that with ocean acidification and pH. And so what we did with temperature is we turned that into an index, and we said if you're below about 10 degrees, it's a little bit cold for toxin production. Optimal would be 10 to 14 degrees. It's decreasing as you get to 20 degrees. And then if the ocean got above 20 degrees, you're really not going to see domoic acid production. And so we're starting to incorporate that into some of our predictive models as basically an envelope approach where we can refine our models. And so taking that same concept and applying it to pH, here's what we get where we use the data from the Santa Cruz Municipal Wharf, my backyard. And so we have pH and we have domoic acid that's being collected basically simultaneously. The domoic acid in this case is taken from spat bags, and so it's an integrated weekly measurement of how much domoic acid we're seeing in the environment. And then I've taken the median pH for that same week. You get the same pattern if I take the minimum pH or the mean pH. And what I want to point out is that generally the domoic acid in Monterey Bay increases as pH is dropping, and we reach a maximum at about a pH of 7.84. The line is the mean of 1,000 bootstrapped runs, and then the gray bar is the standard deviation, giving you an idea of how much variability we have on that. And so we can do this because we do collect these data simultaneously at these sites. And this is just one example, picking Santa Cruz Wharf, because I know that area and I understand it pretty well. Oops, I forgot I had that in there, sorry. And so again, the idea of bringing it back to the lab, this is some really nice work that was done by a master's student in Bill Coughlin's lab for his thesis. And he took pseudonychia into the laboratory and looked at exponential growth and stationary phase and how much total domoic acid you would find as picograms per cell. And what I want to point out is in this right panel, he saw in culture that the maximum domoic acid per cell is at about a pH of 7.8, and it drops off relatively quickly as you go down to a pH of 8.1. And so in this case, it pretty much exactly matches what we see in the field, that we see maximum domoic acid in the field at a pH of 7.84, and it drops off pretty quickly as we go down to a pH of 8.1. And so a really nice example of iterating, going between the field and the lab. We can also do that with mussels. Here is data from Coast Seafoods in Humboldt. You'll notice the probability is much weaker. This is in part because it's a relatively small data set. But again, we don't see a lot of domoic acid in the mussels until we get below, significantly below a pH of 8. And so these are kind of trivial examples, but they show you what we can start to do given that we have these long time series of both OA data sets and HAB data sets. We can start to see, are they at all correlated? Are there connections between these parameters? But that doesn't tell us what's going to happen in the future. And so what I want to introduce is this really cool paper that was published by Sinead Collins and co-authors in 2014. And what she was looking at is what's the difference between short-term adaptation or plasticity and long-term evolution in harmful algal bloom species. And so I've only, again, pulled out the organisms that we find on the west coast of the US, so focusing on California. And just to walk you through this, in these experiments, you grow the organism at high CO2, and so typically these experiments were done at 750 ppm. And then what you do is you see how much faster or slower do they grow compared to organisms that are grown at ambient conditions. And so this plasticity column says that, using linguloidinium as an example, if I grow them in high CO2, linguloidinium grows about 7 times faster at high CO2 than it does at ambient CO2. But we can separate that out by then saying, after many, many generations, we compare it again to the one that was adapted to growing at high CO2 versus the one that was not adapted to growing at high CO2, and we can get at how much evolution changed things. And what we find is that for many organisms, we see that even though there's a short-term positive response, there can be a long-term negative response. And so evolutionarily, they're actually less adaptive. And so that's something that we also need to keep in mind, that these short-term experiments tell us what's going on right now, but they don't necessarily tell us what the ocean is going to look like in the future, because we need to take into account that evolutionary response. And so how do we do better? How do we merge what we're measuring with the OA networks and the HAB networks? I think California is a really good example of this, where they've given it a lot of thought. And so I'm showing you 2 reports. The West Coast Ocean Acidification and Hypoxia Science Panel released a report in 2016. And just very recently, this report came out this month, they followed up with a report on how we enhance the ocean acidification and hypoxia monitoring network. And their outcomes from this, I pulled out a few of their bullets, but they said we need to better connect chemical and biological monitoring, that we need to quantify key species sensitivity and capacity for adaptation, and that we need to assess ecological system sensitivity and resilience by employing an ecosystem modeling framework that explicitly tests predictions against empirical observations and identifies model sensitivities to model formulation and parameterization. What I would like to point out to you is that this is all about ocean acidification. This was not a report on HABs. There's basically no mention of HABs in this. And yet, if I just took those bullets and stuck it into the next harness report, I think anybody working on HABs would completely agree with that we need to have better chemical and biological monitoring, and it needs to be better connected, that we need to understand species sensitivity, and that we need to move towards end-to-end modeling. And so both of these communities are moving in the same direction, but we're not necessarily linking them at the point of implementation, with a few exceptions. And so one example of how we can do that, we have the statistical model called CHARM, which I'm showing on the left. This is based on statistical parameters that was originally described and maintained by Clarissa Anderson. And so it looks at things like ocean color and temperature, and it can give a 3-day forecast of how likely it is that we're going to see domoic acid in California. We're moving with Daniele and others at UCLA to embedding this within the ROMS-Beck model, which is a full physical model that has biogeochemistry in it. And of course, if we go up into the fisheries groups and the OA groups, they're really interested in combining all of this into things like the Atlantis end-to-end ecosystem model, where we can actually start to map out ecosystem structure and pathways and how things are interacting with each other. And so I would argue very strongly that this is exactly the direction that HABs need to go in as well. And that if you're building these models for OA, it's not that difficult to take that same model and to use it to address harmful algal bloom questions. And so using this as an example, here's the sea harm model. And I just want to point out that right now the sea harm model is using remotely sensed data. And so we're using temperature, we're using chlorophyll, we're using ocean color, but we do not have things like nutrients in it because we can't remotely sense nutrients, and we don't have a network of nutrient sensors that cover the entire offshore region of California. But we know that if we had nutrients, we could put them in there and it would improve the model. And so there's been several papers showing that from a statistical perspective, adding nutrients definitely improves our ability to predict things like toxicity in Pseudonychia, and it would also improve predictions of any other HAB. And so where we're going with this is we're starting to do that within that Roms-Beck model. We're taking this conceptual model where we actually start to model the physiology of Pseudonychia, and we include nutrients, so we can move away from purely statistical models and we can start to get to physiologically driven models. And the way we're doing that is by incorporating that into the Roms-Beck model. And so Roms-Beck is an ocean physics model that already includes groups of phytoplankton, different types of nutrients, but it also has OA parameters. And so it has oxygen, it has pH. And so what we're working on is we have an EcoHead project that's going to embed this Pseudonychia model within Roms-Beck. And so what's super exciting about that is we'll actually be using the same model that the OA people are using, but we'll be addressing the HAB question. And because we're doing that, we can start to interrogate the model and see how those parameters are interacting. And so here what we're looking at is a really high resolution run where Daniele and his colleagues basically turned on and turned off nutrient supply from wastewater discharge. And so what you're seeing here is when they add the nutrients from the wastewater discharge, they get a big increase in primary productivity, and that drives down the pH and it drives down the oxygen. And so we can start to see the linkages and the interactions between eutrophication, between OA, between the potential response to HABs, even though in this particular example, it's still just chlorophyll. Another example from their model output, and so they have OA parameters for the entire West Coast. On the left here, what we're looking at is their modeled estimate of pH, and so the sort of light orange-yellow color is low pH, the purple color is high pH, and so this is during the spring in the top, and then during the summer in the bottom, and we see the strong seasonality driven by upwelling. But what I want to point out is there's some hot spots that show up where we have lower pH, and those hot spots are the same places where we're seeing hot spots of domoic acid. And so again, going from the model, going back into the field and iterating back and forth, we are seeing correspondences between what's going on in pH and ocean acidification and what's going on in harmful algal blooms. And so I think there's a lot of promise here, and we're really just sort of scratching the surface here. We're looking at correlations, but haven't really gotten to a mechanistic understanding of how these interactions are occurring. And so just to wrap up, what I really want to emphasize is I think we have to be a little bit skeptical about looking at a half dozen analyses and saying that's what's going to happen in the future world ocean, because we know there's a lot of plasticity in these experiments, there's a lot of strain differences, there's a lot of species differences, and we need to start building up data sets that can account for that. And so a big question is, how representative are the existing experimental data on HABs and OA? I'm not at all criticizing the papers that are out there. I think the papers are really nice analyses, but it's a small data set that we're using to extrapolate to the entire world's oceans. Second, How will those HAB organisms respond on short and long-term scales? We have not very many experiments on short time scales because they're difficult to do. We have even fewer experiments on long-term evolution time scales. Most of those on our coast have been done by Avery Tatters, and I think we really need to see more of those experiments. And then, of course, it's complicated and messy, but the next step beyond that is we really need to start looking at those multiple stressors and seeing what if we're changing all of these things at the same time versus doing one of them at a time. My third main point is that we really do need to link this to higher trophic levels. I haven't talked a lot about that today, but we know there's an entire community that's looking at ocean acidification and stress responses in Dungeness crab and whether there'll be recruitment success in shellfish. And that has to be factored in as well. What happens if you're already stressed for OA and then you're exposed to 3 or 4 different types of harmful algal toxins? I would argue that we pretty much don't know what the response to that is, and we're probably going to need Atlantis-type models to get to those answers. And then lastly, my plea, which I've said this several times before, but I do think we can better coordinate between the HAB and OAH communities to take advantage of this overlap. No, we're not measuring exactly the same thing. No, we don't all need exactly the same parameters, but California just committed over $1 million to start tracing nutrient sources to look at OAH. And again, that is exactly what we need for HABs. We would love to know what the nutrient sources are and link that to harmful algal blooms. The problem we have right now is our communities are sitting next to each other, we're observing next to each other, but we're not actually funding together. We're not jointly coordinating these programs. And so I think we could easily improve on that and make huge strides going forward by just simply opening up the lines of communication and talking to each other a little bit more about how could we best collect the data so that it's useful for all of these groups at the same time. And so I'll stop there and would be happy to answer any questions that you may have.
Thank you, Raphe, for your presentation and also for your recommendations. We will open the conversation for questions now, and to do that, please type your questions in the chat box. Maya will read the questions from the chat box, and we'll try to unmute speaker or questioners as the questions are read so that you can have a follow-up conversation with Raphe on your particular question. If others want to comment or contribute to the discussion of a particular question, please raise your hand and we will try to unmute you if time permits for discussion. We ask that everyone confine their discussion to 2 to 3 minutes per question so that the speaker can address all questions that come through. All of the questions will be recorded, so if we don't get to your question today, we will compile it for discussion at the online workshop in August. And we are also asking that all participants complete a short Google form. The link can be found in the chat box at the end of the questions. Jen will enter that. And the form is also a good place to add any comments that we don't get to today. We're using responses to help guide our discussions at the August workshop to inform future research funding directions for ocean acidification and harmful algal blooms. Maya, what is our first question? So I'll take over if we are having issues with audio, but Maya, you're currently on mute if you are able to come off of that. Okay, I'm going to read questions, and then if she is able to join us through audio, then she can join as we go forward. So the first question is from Maria Kavanaugh. And she mentioned that early in the talk, Raphe mentioned that the temperature optimum of the domoic acid cumulative probability analysis was about 12 degrees C. Off of Oregon and perhaps too in Northern California, that temperature might indicate downstream from upwelling, where upwelling actually would be much cooler and further left on the CPA curve. While nuanced, the time-space delay is important. pH would be lowest in the newly upwelled water near shore, while nutrient stress and competition might occur further offshore. The question is, have you tested some of the interpretations of CPA analyses using the ROMS model to investigate the cross-shelf spatiotemporal decoupling of ocean acidification and nutrients with the upwelling?
That's a great question, Maria, and I appreciate that. And we have thought about that, and we've looked at that in a couple of ways. And so the CPA is a convenient way to distill down a whole bunch of data into one graph. But we've done similar analyses looking at exactly that, looking at the spatiotemporal interactions. And you're right, there are subtleties in there. I would refer particularly to John Ryan's paper from a few years ago, where he showed that the maximum toxicity in Monterey Bay was actually when temperatures returned to normal, rather than being during the intense upwelling period or during the heat wave. And so that's a really good example of some of the subtleties. Have we done that with ROMS? Not yet, but what we are doing right now is we're taking all of those factors and we're using GLM and some other statistical logistic regression approaches, as well as CPA, to see if the ROMS data gives us patterns that make sense compared to our field observations and the laboratory observations. And so that's absolutely the direction that we're going in. And I completely agree that there's a lot of subtleties in here, and that's where the really interesting science actually is.
Thank you, Raphe. There is a partial question from Terrence Fleming. Terrence, if you could complete the question in the chat box, that would be helpful. But he asked if you could talk a little more about the timing of your EcoHAB work to the Ronsbeck model, is what it looks like.
Sure. So our EcoHead project, and so Daniele Bianchi and his other colleagues have been using Ronsbeck for a long time to look at the biogeochemistry of the California current. And so that same modeling framework is being used to look at pH and OA, but also the role of anthropogenic nutrient loading. What our EcoHAB funding has provided is an opportunity to put a pseudonychia-specific model within Ronsbeck and to show what could we do if we actually had full fields of things like nutrients that could drive this more sophisticated model. And so we're about a little over a year. Daniele actually just submitted the report, or 2 years, maybe 2 years, coming up on 2 years into this. And so where we are right now is we've been developing a model that can, or a module that can be inserted into Ronsbeck. And so we've been developing the physiological model using batch cultures and chemostat data. And we're now moving towards testing it first in a 1D and then a full 3D setting. And so we're hoping that within the next several months, we'll actually start to be able to generate full three-dimensional, physically, biologically coupled observations of what we think pseudonychia would be doing in that model. And then, of course, that's when it gets fun, where we can turn on and off nutrients, we can change the temperature, we can see how it changes the pH.
Thank you. Our next question is from Beth Turner, and she asked, do you think that models should be incorporating both harmful algal blooms and ocean acidification into one overarching model, or can separate models for HABs and OA be compared and gain some understanding?
It's another great question. I absolutely do think that separate models can be compared as long as they're consistent in their drivers. However, I think the Ronsbeck one is a really good example. You know, the HAB component of that did not drive the development of Ronsbeck. That was driven by interest in biogeochemistry. But for a small additional investment, you can add HABs to that existing model. The interest in nutrient sources is being driven from a question of how are anthropogenic nutrients driving ocean acidification, but you don't have to pay anything at all to take that data and to ask the same question about HABs. And so I think there's synergy there where you can add HABs to existing models, but I think you could also just take the output from that model and look at it to understand HABs. And where I think where we've been lacking is having good conversations between those groups so that we both know what's available and how that model is actually running and what the pros and cons and the potential pitfalls are of doing that sort of analysis.
Thank you. We're going to try Maya's audio one more time, and she's going to take over with the next question.
Yes, the next question is from Francis Chan. He asks, it seems that future HAB distribution will be a function of physiology, evolution, and ecology. Is there any guidance on which of these may be the source of our greatest uncertainties and thus to tackle most intensively?
I will give you my opinion, and I'm sure there's probably many other opinions on this webinar. I think we have a pretty, for some organisms, we have a pretty good handle on the physiology. There's been a lot of really nice work done on that. We have a good handle on the ecology in the modern ocean. I think there's very, very little known about how these are going to be translated through the lens of evolution. And so are these organisms going to evolve? Is there going to be a response on the long term? And so that's where I think, I'm definitely not saying that we're done with physiology and ecology, but I do think we need to start bringing more of the evolutionary responses into our experiments looking at sort of the modern ocean.
Thank you. And our next question is from Stephen Wilhelm. He stated, Raphe, you keep mentioning nutrients, but are you talking about nutrients at some prior time to the bloom or at the time? There is a challenge of having to wait for the cells to grow in all this. Most people measure both at the same time, but when you do the nutrients are as much the residual of a bloom event as they are the driver. I believe that was more of a statement than a question, and we have another participant typing another question.
Well, while they're typing, I'll just comment to Steve. You're dead on, of course, and I think it also highlights Maria's point that there's subtleties that I've glossed over. And so I do mean both. I mean, what were the nutrients that you started with, and then what's the nutrient stress? And that's why we want to go to a physiological model rather than a statistical model where we can actually start watching those time-varying changes.
Thank you, Raphe. We are waiting to see. I saw some people typing. We may have additional questions. And if anyone who has not asked something at this point wants to chime in, they are welcome to. And that may be it. So thank you, everyone, for your participation today. And thank you, Raphe, for a great presentation and for sharing your recommendations with us. And everyone else who, oh, I do actually see Jen Wolney has asked a question to follow up on Stephen's. She asked, what would you recommend as the minimum frequency for monitoring nutrients?
I'm going to give you two answers to that. As a HAB person, I would say the minimum frequency is you should be measuring nutrients whenever you're measuring HAB parameters. And so if you're doing cell counts or you're doing toxin, you should also measure the nutrients. But beyond that, nutrients is kind of a generic term, and I would strongly advocate that we should be measuring the full suite of nutrients, including ammonium and urea. And we should also be taking the OA parameters at the same time if they're not being taken. But beyond that, kind of a more philosophical point is, I think what's really driving a lot of the exciting things in our community right now is the ability to start measuring things at relevant ecological scales. And so we can measure temperature at second or sub-second scales, but now we have things like IFCBs where we can get phytoplankton composition every 25 minutes. And we can start to do the same thing with nutrients. And so if we have the capability to put something like a SUNA out there and get really high resolution nutrient data, then then that's just more information. And if you don't have that, you don't know what you're missing. And so I would advocate that, it would be fantastic if we can move to more and more automated nutrient sensors that are measuring on time scales of, maybe half an hour to match something like an IFCB. But at minimum, you should be measuring nutrients every time you're measuring a HAB parameter.
Great advice. Thank you very much. And I think that is it. I haven't seen anyone else typing in the chat box, but I wanted to remind all participants, and particularly the participants of the workshop in August, to please remember to complete the Google form. I think Jen will add that back in the chat box. Hopefully there will be a link, and if not, then it is on the workshop page that was shared at the beginning of the chat. There is a workshop website that you can access, and there will be the recorded presentations and also a link to the forms for each presentation that you can fill out to provide input that will be considered as part of that workshop and that Maya will be collating as part of her summer project. Thank you, Raphe, and thank you everyone for attending.
And please join us on Friday when our speaker is going to be Hans Pearl.
Yes, Beth, thank you for that. Friday at 1 o'clock, Dr. Hans Pearl will be presenting on acidification and harmful algal blooms in estuarine waters. That's 1 o'clock Eastern Time.